Thank you, everyone, for coming to this panel discussion. It's a really interesting topic for me for a long time and intriguing in many ways. So the topic of our discussion is review of genetics research protocols involving American Indian tribes, a researcher, tribal nation, and IRB perspective. I am a project director at Sanford and also a member of the Sanford IRB. Today I have the pleasure of introducing three panelists who play a key role in the field of research regulation and have been conducting research for several years in the Northern Plains communities. Dr. Dewey Ertz is a member of the Cheyenne River Sioux tribe and a practicing psychologist in Rapid City. Dr. Ertz. He's also the chairperson of the Aberdeen area IRB. He's been in clinical practice at the doctoral level since 1977 and has worked in private residential Indian school and in community health for Cheyenne River Sioux tribe. His practice includes providing evidence-based intervention techniques and cognitive behavioral methods to a wide variety of patients, including children, adolescents, and adults. Our second panelist is Ms. Cecelia Big Crow. Ms. Big Crow grew up on Pine Ridge and is an enrolled member of the Oglala Sioux tribe. She's been involved in Native research since 1997. Cece was instrumental in establishing the Oglala Sioux Tribe Research Review Board. She works for the University of Colorado in the Center for American Indian and Alaska Native Health Program. And our final panelist is Dr. Lyle Best. Dr. Best began his professional career with the Indian Health Service in 1977 and served as a clinical director of an IHS hospital for 16 years. Since 2000, he has served as principal investigator for the Strong Heart Study, the largest epidemiologic study of American Indians ever undertaken. He's also the PI for the pediatric asthma study as part of the CRCAIH. Please join me in welcoming our three panelists today. So we'd like the session to be interactive, and this is your opportunity to ask those tough IRB questions. So we'll focus our discussion today on medical genetics, because as we know, genetics is a really broad term. So medical genetics as a direct study of determinants of disease. Just a few tips before we start the panel discussion. I'm going to pose the question to the panelists and have each one of them respond. So please hold your questions until each of the panelists have had a chance to respond. If you have a question, raise your hand and we'll bring a mic to you. Or if you do not want to be identified, you can write a note and pass it along and we'll read your question anonymously as well. Okay. So can I get a show of hands for people that have been a research participant or conducted research with human subjects? Okay, great. How about those who have served on an IRB or currently serve on an IRB? Awesome. And how about those that have interacted with an IRB? Okay, so you can feel our pain. All right, so my first question is, from a community research review board or an IRB perspective, what are the key elements to consider when reviewing genetics research protocols with American Indian communities? Please provide specific examples. I'd like
Whoever wants to start.
Yes, whoever wants to start.
I'm assuming that this thing is on. Everybody can hear me all right?
No.
How do we turn it on?
It's got a little switch at the top.
Yeah, little switch at the top.
Now am I on?
Says on. There we go. I just have to get there in the right spot, right? Okay. I think the first thing that you have to do from an IRB perspective is realize that genetic research by definition is more than minimal risk. So it's going to take more effort and more time to go through and deal with consent forms and making sure that everything is streamlined in a proper way. You also have to be careful about your research participants because there's certain guidelines that you have to look at when you're dealing with high-risk kinds of participants, especially children, prisoners, people with developmental disabilities, and females in general. Those are all things that you have to look at in greater detail and make sure that you're working with. The other thing to keep in mind that as an IRB, as being a chairman of an IRB for a long time, we do not intend to be your enemy. Okay? We may end up being your enemy, but that's not our intention. Okay? We are required in a very real sense, especially within the Indian Health Service, to make sure that you have everything procedurally accomplished in a reasonable way and that you have the right kinds of collaborative individuals that are going to help you implement your protocol. We will look at the risk factors as a primary concern and protection of human subjects. So if you have any questions about that, you want to go back and look at that data, which is well published, and also consider that it goes back to the Nuremberg Code after the Second World War. And that line of what an IRB should be looking at is pretty strongly established and something that you have to work with. Another thing that you have to make certain of is that you are doing all the correct procedures for informed consent for participants or assent if you're dealing with children. Those are the two areas that tend to be the most critical and the most technical to work with, and the things that we're probably going to look at the most because that has a direct application for protection of human subjects.
From a community tribal research review board perspective, and I'm no PhD, so I just want to get that out there right away. And I was a little nervous coming up here because I thought, oh my gosh, there's so many smart people, and I want to first ask for forgiveness for my elders for speaking before you, because I know a lot of people in this room carry a lot of intelligence and a lot of experience and things like that, so I don't want to offend anybody by speaking here today. But I'm very honored to be up here. Glad to see all of you guys here. From a community tribal research review board perspective, I think genetic research at least in our community that I work with, we really need to look at how the benefits outweigh the risks. We also talk about shelf life, how long we're going to allow people to keep this material, whether it be genetic or intellectual property. How long will allow the researchers to analyze this material? We have, which Lisa, Dylan, and I will share later on, a lot of stories about different research studies that we've heard about or that we've also experienced, that we've learned along the way, different processes. Because I think we're kind of blazing a new trail by having the Tribal Research Review Board. Some of the other things that we want to look at is whether or not our cultural practices are being respected when we talk about genetics, genetic research. So those are some of the areas that we look for. And further in that data analysis, whether or not you propose to do something that's within the realms of the original consent. So if you are doing secondary data analysis, we really scrutinize and analyze that, whether it's covered under that consent or the original one.
Okay, I guess I agree with all of those things, and I guess I'd just bring maybe two additional comments forward. One is that I think that there's been a sort of a false distinction in many ways between what is genetic testing and what is other typical testing when you go to the clinic. And we've been doing genetic testing for decades in terms of blood type and so on. It's very non-controversial, very routine. Been doing newborn screening testing for years and years. It's also very non-controversial. One might even say it's hardly even consented by the new mother for her newborn. And that's not to say that those are good things. Perhaps more should be done in terms of consent or education around those areas. But at any rate, I think there's been sort of a false dichotomy between, oh, there's genetic testing and genetic research, and then there's everything else. And I think the line is not nearly so distinct between the two. The second point I'd make just briefly is that in terms of genetic, can you still hear me? Okay, in terms of genetic testing, one of the problems we have currently is that much of what we're learning in terms of genetics is still very, very, very preliminary, and there's relatively few genetic variants that we are testing for that have any direct clinical application. It's just really quite unusual that much of the research has any direct clinical application. So therefore, when we do testing, the question comes up in terms of giving results back to the individual participant. If we check your cholesterol as part of a long-running cardiovascular study or something, we'll let you know what it is. We'll give you advice if it's too high. You can seek referral and so on. But the vast majority of genetic testing we do, we really don't have any way of giving that back to the individual participants in a useful way. And we also run afoul sometimes of the difference between a research laboratory test result and a CLIA certified laboratory test result. We're technically not supposed to give back research laboratory test results unless they are done in a clinically certified laboratory. And personally, I don't really agree with that, but that is a problem we run into occasionally. So just something to be aware of if you are on an IRB reviewing a genetic protocol.
One of the questions that I was going to ask in a comment is that with the OSD IRB, you have the investigator come in, and there's an open dialogue about the study and the risks and the benefits. And you, as the IRB, have found that very useful. So I was just going to ask Dr. Ertz, does the Aberdeen area at all do that, where there's a dialogue with the investigator about the study?
We don't do it routinely. We have done it. One of the things that you need to understand is that we're spread over more than 5 states as a committee, so our meetings are not face-to-face. I probably can recognize about 1/3 of the people in our IRB by facial contact. I recognize them all by voice contact because that's how we meet. But we have had investigators come in the telephone calls and talk about it. We've also had people sometimes go out and do direct contact with people who are submitting research proposals, especially if there are issues. One example is a protocol where there were some visual activities used or posters used for part of the consent form, and I went to actually look at that and watch how that consent form was implemented. So if that's necessary, especially when we're dealing with consent, that is something that we will try to do or work with so that we understand exactly what you're telling us on your protocol. It may be whoever is the closest to go. It's not something that we can really work with. As an IRB, we're mostly all volunteers. We don't have any connection or budget or anything like that to do travel with things. And we're certainly able at times for somebody to meet with you as a researcher if there are issues or questions that you have that we need to address in some way. And I think the other thing to keep in mind is a good IRB is a collaborative effort with you as a researcher. We want to foster good research as part of our role in making sure that things are followed correctly. And we may ask some questions you don't like of us at times, and you may have to explain some things you don't think you should have to explain the second or third time. But really, that's the idea, I think, in making this work well, is it has to be a partnership. You're the researcher, we do the IRB work, and we are responsible then back to the tribal entities to ensure that that's done in a consistent and open manner, again, to protect subjects. Another piece that you have to understand about the Indian Health Service IRBs, if you haven't run into it, all your publications come back to us for approval. That's a different role than a lot of IRBs have. So when you're ready to submit a publication, even if it's a abstract at a convention or a PowerPoint presentation, those come back to us and we will read through those and look at them and make sure that there's nothing that we would consider detrimental in some way or misstated in some way from a cultural perspective or from a community perspective. So you have that added issue at times that you have to consider. It can make the publication process a little bit elongated. I think we're getting better at trying to keep that in a reasonable way, but that's still part of our requirements, which is not a usual IRB function.
And can I just add, as part of the Community Tribal Research Review Board, the interaction has been beneficial both ways. I've heard a lot of PIs, principal investigators or researchers, that tell us back that we really enhanced their research because they didn't know a lot about our communities, and we were able to go to them and say, when they come in and present their work, to be able to say, well, so-and-so has done research in this area already here in our community, go and look up that work, and it'll help guide you to making your work a little bit easier so you don't have to reinvent the wheel. And that's one of the reasons I think we started the Tribal Research Review Board. I mean, there are many reasons, and we'll go through that. But one of them was that we started seeing a lot of research that been done 50 years ago, and then they're coming around doing the same kind of research. And we thought, you know what, this is kind of crazy because it's going to find the same thing. And we all know pretty much in the community that we have these kind of issues. We don't know what the extent of those issues are. So the work between the PIs, the investigators, and the Tribal Research Review Boards is more collaborative, I would say. And I know IRBs are really collaborative too, but we are able to give recommendations and to say, you know, these are the ways that we do these things in our community. If you want to interview a woman about her sexual experiences, you need to be a woman interviewing them. So we try to make it gender specific because that's our cultural way. We just would like to see the research mean something to our people because we've been researched. We always say that we've been researched to death, which it feels like we have, but in the general scheme of things, there's very little known about Native American health or American Indian health and our disparities. But so when we say that, we're feeling like people would come out here, survey us, interview us, take samples, take cholesterol readings, and we never really got anything back, or it didn't improve the health that they were looking at. So we want to stop that business, and we want to make it research mean something for us.
Before I go on to the next question, did the audience have any questions?
Okay, yeah, my birthday's in May. I know you guys were wondering about that.
So Dr. Best, I'm going to ask you this next question. From an investigator perspective, what are the elements to consider in protocol design for a genetics research protocol?
Right. There's really quite a few. I think first of all, just like with any research, you need to be focused on, you know, what is your research question? Are you going to be able to answer that? You know, do you have adequate power in your study? You know, what is the study design that's going to do the best job, and so on. But okay, bring this a little closer. I think this will be okay. So then beyond that, we discussed this a little bit at the executive committee meeting yesterday afternoon in terms of people are always interested or frequently interested in you know, what is going to be the disposition of my samples, my data, and so on. Who is going to be caretaking them? How are they going to be cared for? Are they going to be identifiable? Are they going to be available to other researchers in an anonymous way that you may not be aware of at this point, and so on? A lot of people don't seem to be aware of the fact that anonymized samples are not really they're not subject to the original consent, generally speaking, and so there is opportunity in some areas, some ways to use anonymized samples that the original participant wouldn't have anticipated. But I think those kind of things need to be considered. You need to consider, very importantly, what is the community expectations, and what do they understand about what is going to be done, and are they really in tune with what the whole purpose of the project is? I think that's really critical. I guess one of the things that's maybe come a little bit more naturally to the work that I've done is that I was a practicing physician with Indian Health Service for a little over 20 years. I stayed in the community over 30 years there. I taught at the tribal college for another 10 years. And so there was a lot of familiarity between myself and community members. And so there's a certain amount of trust that develops as a part of that. And I think that's a really important, I'd like to see that in more research. And I know we all tend to be specialized in our society, and investigators have very focused expertise that they bring, but I'd like to see more and more community involvement in research, not just from the standpoint of the participants, but from the standpoint of the investigators. And it seems to me that there are lots of clinicians out there that should be given some additional time to expand their professional work and to include some research. And I think there'd be a lot of benefits both for the communities and the investigators that way. That's just my current soapbox. I'll apologize for throwing that out there.
Do you think people are more guarded if they see, so for example, for a sample collection, if there's saliva versus blood for DNA?
Yeah, that came as a surprise to me. In our the study at Turtle Mountain Community College, we're looking at the genetics of preeclampsia, influences on preeclampsia, and we initially began by doing capillary blood draws on the FDA paper. It's called Save the DNA, and it's a nice way to preserve the DNA. When we first started doing that, I wasn't really aware of the technical advances in collecting and saving DNA from saliva, and that came about a little later on. And it turned out that we needed that salivary DNA was a little bit easier to deal with in another collaboration we're doing, and we had to go back and reconsent everyone. And we had individuals who just said, "No, you can have all the blood you want, but you're not getting my spit." And I don't know. Maybe there is some